So now we're going to look at CRISPR-Cas9, and this is a way to edit DNA in living cells. And the ramifications of this ability and the potential for that this technique gives us is absolutely insane. So the various ethical issues that we need to now start looking at is when we think about changing the human genome, is that do we limit that to somatic cells, cells that won't be passed on to further generations? Do we modify germline cells that will permanently change the entire ancestry of or descendants, the DNA of the descendants of a particular person? So there's lots of stuff going on here. And in fact, the idea that this is such an important technique has, I don't like that. So now we're going to learn about gene editing via CRISPR-Cas9. And what CRISPR stands for is clustered, regularly interspaced short palindromic repeats. An absolutely massive breakthrough that allows us to edit the genome of cells, of living cells. And the consequences of this technique we're still sort of dealing with and understanding and trying to figure out how we're going to apply it. And it's so important. It won the Nobel Prize this year in 2020, and in the Nobel Prize in Chemistry by Emmanuel Charpentier and Jennifer A. Doudna. Now, before we start talking about CRISPR-Cas9, the one thing I think many videos that describe this and what many articles that talk about it, what they fail to mention is that what CRISPR-Cas9 does is absolutely essential on something that already occurs in all of our cells. The idea that naturally all of our cells have a way of dealing with double-stranded breaks that occur within our DNA. So double-stranded breaks occur. These are a form of DNA damage that can occur for all sorts of reasons. And because it just naturally occurs, a system has evolved in all of our cells that deals with repairing those breaks. So this slide simply shows you two of the endogenous ways in which our cells can repair double-stranded breaks. Now, I want to stress here, this is totally normal. These repair systems exist in all of our cells. This is 100% completely independent of what CRISPR-Cas9 is. However, our use of CRISPR-Cas9 absolutely depends on these endogenous normal systems of double-stranded break repair. Okay, so something happens within our cells and it breaks DNA in both strands. And so there's two ways in which our cells can deal with this. One is non-homologous end joining, which is to simply just find the ends here and try to attach them together as best as the cells can. Now, that form of double-stranded break repair oftentimes results in mutations. Now, most of the time, a double-stranded break is not going to occur in the middle of a protein encoding gene because if you'll remember, it's only like 1.5% of our genome. So most of the time, it's not a big deal if a mutation is incorporated as a result of this type of double-stranded break repair. Now, the more complex form of double-stranded break repair, the form that is more often occurs without mutation, is called homologous recombination, where what happens is the cell has proteins that detect that there's been a double-stranded break, and then it sort of eats away one of those strands and then uses, remember, we are diploid organisms. For every region of DNA, we have two copies of it. And so the cell uses that other copy of DNA as a template to repair this portion of the DNA. Okay, that is called homologous recombination. And so that now you have repaired that double-stranded break, and you've repaired it more likely without mutation that comes along with the non-homologous end joining. Okay, and it's this particular way in which cells or double-stranded breaks are repaired that becomes important when we're talking about editing genes, fixing genes. It's this method here when we're talking about breaking genes using CRISPR-Cas9. Okay, so these two mechanisms, totally endogenous to all of our cells, has nothing to do with CRISPR-Cas9. So what does CRISPR-Cas9 do for us that we couldn't do already within our cells? It's right up here. CRISPR-Cas9 offers us a way to incorporate double-stranded breaks in DNA at very specific places. And then we rely on our cells' natural ability to repair those in these two different ways. That's what CRISPR-Cas9 allows us to do. Now, CRISPR-Cas9 system was originally discovered as an adaptive immunity system in prokaryotes. So if we have a bacterial cell here, well, bacteria, they often are the victims of viral infections just like you and I are. And they have a way to protect themselves against that. So when a bacteriophage, or a virus that infects bacteria, when it infects bacteria, it injects its viral DNA into the bacteria, and then the bacteria, what they do, if they've never been infected by this virus before, they have a way of copying this viral DNA into their own genome. And that's what are these spacers. So these spacers here, these are sequences of DNA that are the same as various viruses that they've been infected with. And then these repeats here separate each one of these spacers. And we'll get to what's important about those repeats in just a second. Okay, so now let's look at a virus though, let's look at a bacteria that has already been infected by a virus that they are being infected by again. So here is the virus. It injects its viral DNA into the cell, and now the cell kicks off its immunity system. And what it does is it transcribes all of these repeats and spacers. Remember, the spacers are going to be the same DNA as, at least some of these spacers are going to be the same DNA as this viral DNA that's been injected into the cell. At the same time, a protein is produced called Cas9, and tracer RNA is made. The Cas9, the tracer, then bind to very specific regions on this giant RNA that's made here, such that we have the Cas9, we have the tracer that then attracts an RNase, and they sit here such that on top of the tracer is reverse complementary to these repeat sequences that exposes in just the perfect place that viral DNA, that spacer DNA, the viral DNA right there. And then this RNA cuts right there, cuts right there. So what we end up with is a mature CRISPR-Cas9 tracer RNA complex. This is RNA that is reverse complementary to some viral DNA. This is the spacer that has the guide strand on it. And this is the Cas9 protein that has enzymatic ability to cut double-stranded DNA. How does it work? Well, since this is reverse complementary to viral DNA, it binds to the viral DNA, and then the Cas9 protein cuts a double-stranded break in that viral DNA. And now that viral DNA is no longer intact. It can't encode the proteins necessary to make the virus, and so this cell has protected itself against the infection. So what we've done is we've got the gene that encodes the Cas9 protein. We've got the gene that encodes the tracer RNA. And what we incorporate then is our, this spacer sequence, the sequence of the DNA that we want to target. And we put those into expression vectors, and we transfect those expression vectors into cells. So here's one transfection vector that has the gene for Cas9 and has a space where we can insert our target sequence. So wherever we want to cut double-stranded DNA, we're going to put that sequence here within our expression vector. And then we have another vector. This vector here is going to be our donor DNA. So if we want to do more than simply break a gene, if we want to take advantage of homologous recombination to replace a gene and repair a gene, then we have to supply our own donor DNA, the DNA that's going to act as a template to repair that double-stranded break. And that is the job of this right here. So we're going to insert within this expression vector our donor DNA, how we want that affected gene to look after this whole process is done. So then we take these two vectors, recombinant plasmids, we transfect them into cells, and this is what will eventually happen. Here is the Cas9 protein. Here's the guide RNA that is connected to our target sequence that is now reverse complementary to and binding to our DNA sequence. That causes a double-stranded break to occur right here. And now, since we have included our donor DNA right on that other expression vector, that donor DNA then is going to be used as a template to repair this double-stranded break, thus replacing what gene used to be here with whatever we had in our donor DNA there. We can do that in human cells, we can do that in model organisms, we can do that in bacterial cells, we can do that in pretty much any type of cells. And so the ramifications for this is we can start to edit human genomes. Now, if we edit genes within somatic cells, like skin cells or liver cells or so on and so forth, that will fix those cells within an organism, but those fixes won't be passed on to later generations. If we edit DNA within sperm cells or egg cells or any cells that are going to give rise to sperm or egg cells, then those edits are going to be passed down from generation to generation. That's called germline editing. And that's the big question now is, do we do that? Do we step over that line and start editing DNA that will then be passed on from mother to offspring and so on and so forth?